Hello, and welcome to Arcadia University's BI327 Histology course. This is the lecture series on epithelial tissues, and in part 2 of this lecture, we're going to take a look at cell membrane domains. Now, the important thing to remember with epithelial cells is that we've got these cells in contact with one another, and they're going to be found forming a sheet along free surfaces of the body. And so because they're going to be essentially forming this boundary, forming this layer, forming the sheet along free surfaces of the boundary, they're going to be a polarized cell. Whether we have one cell thick or many cells thick, at some point this epithelial lining is going to be adjacent to that open space, that biological space, whether it's inside of the body or outside of the body. And at the opposite end of that, we're going to have the epithelia anchored to the surrounding tissues, anchored to the underlying connective tissue, and anchored to the rest of the body. So the presence of these different regions in relationship to the cell is what makes this polarized. And so if we take a look at a generic epithelial cell, what we're going to see is that they're going to be an apical domain, an apical surface, which is basically the apex or the top of the cell, and that's going to be the portion of the cell which is going to be adjacent to that free surface. At the opposite end of that cell is going to be the base, referred to as the basal portion of the cell. And that's going to be the bottom of the cell, and this is where it's going to be anchored to the basal lamina, anchored to the basement membrane if we're looking at it in terms of light microscopy, but basically anchored to the rest of the body, anchored to the connective tissue that's going to be underlying it. So we've got a top of the cell, we've got a bottom of the cell, and then we're going to have the lateral portion. The lateral are going to be the sides, which are basically going to be adjacent to all of those neighboring cells. Again, keep it in mind that the epithelial cells are going to be in close proximity to one another, and they're going to be in contact with one another. And so we're going to look at those interactions that are occurring with the cells in relationship to one another. Now, we're going to focus in on the lateral specializations first, and we've got to come up with some terminology so that we can understand what it is we're talking about. The first term we're going to use are zonula. Zonula are going to be belt-like connections, which in essence are going to be wrapping around the entire cell. And in doing so, it's going to be essentially 360 degrees around the cell, all the way around, whether it's a round cell or kind of a polygonal cell. It's going to be wrapped all the way around it like a belt. In contrast to a zonula connection, we can have macula connections. And macula connections are spot-like connections. They're disc-like connections that are at specific points along the membrane. And so you can think about them in a non-biological sense as like spot welds or like rivets, which are going to be important for holding the epithelium together. Now, while we're talking about this terminology, I also want to introduce the term gap junctions. Gap junctions are going to be similar in that they're going to be little spot-like connections, but they're going to be very, very different in both structure and function to the macula connections that we're going to be talking about. So if we take a look at the lateral specializations, probably the easiest way to look at this is to look at the terminal bar. And the terminal bar is a light microscopy term for two substructures within it that can be identified using electron microscopy. This is going to be the zonula occludens and the zonula adherens. Now, keep in mind that with the zonula connections, again, they're belt-like connections are going to be running all the way around the cells. The zonula occludens towards the top is going to be a tight junction. It's going to have about a 10 to 15 nanometer space between these cells, and it's basically going to be like a Ziploc bag. When the two portions of the zonula occludens come together, it's like yellow and blue make green. We're basically going to seal off that epithelium so that nothing can pass through it, so that anything that's going to be going across this epithelial lining is going to have to pass through these cells. Lower than that, along the lateral surface, is going to be the zonula adherens. Again, zonula for belt-like connections, but adherens, this is for adhering or connecting the cells together, holding them together. And we're going to see that this is going to be essentially there to essentially strengthen that connection between the cells. A little bit wider, about 20 to 90 nanometers in space between it, and it's also going to be anchored into the cytoskeleton. It's going to be anchored to actin along that cytoplasmic side. So if we look at this in a little bit higher detail, looking at it in essence in artist renditions of this, the first we've got is the zonula occludens, that Ziploc type connection. It's going to form the tight junctions again. And so we're going to have proteins like occludin and claudin, which are going to create a diffusion barrier. It's going to prevent things from essentially getting from that apical space and then diffusing down between these cells. It's going to form that tight junction, form that barrier, so that anything that needs to diffuse either from the lumen into the rest of the body or from the body into that lumen is going to have to be passed through these cells, regulated by these cells, and it may be modified in some way as it's passing through the epithelium. Lower down along the side of the lateral side of these cells are going to be the zonule adherents. Again, a belt-like connection that's going to be wrapped around these cells, but instead of the occludens, like that ziplock connection, we're going to have the adherents. And the adherents are to adhere the cells together, to hold the cells together. And so we're going to have another set of proteins that are going to be there on the extracellular surface of the cells. And these are going to be things like the cadherins. And they're essentially going to match up with cadherins on the neighboring cells and essentially clasp one another and hold the cells together. Now keep in mind that there's going to be a lot of stress on these epithelial linings, and we want to keep these cells together. We don't want them to pull apart. We don't want the epithelial lining to pull apart. We don't want the cells to be lost. So there's going to be a lot of stress associated with it. And if we just had this belt around these cells holding them together, we run the risk of really damaging these cells and damaging the epithelium. So what we're going to see is that these zonule adherents, these connections, are going to be transferred internally, again by protein connections. Proteins connected to the cadherins on the outside through these zonule adherents are going to be anchored to the cytoskeleton. In this case, they're going to be anchored to the actin. And what this means then is that the stresses along that belt-like connection of the cell are going to be distributed across the entire cytoskeleton. So they're going to be distributed across the entire cell. And in doing so, it's basically taking the stress and spreading it out and basically connecting it up with those neighboring cells. And so instead of pulling on one specific little cell, if you're pulling on this, you're going to be pulling on all of the cells. They're going to be attached to one another through these zonule adherents and the connections within the cytoskeleton. The next thing we're going to look at are going to be the desmosomes. And again, these are primarily on the lateral surfaces, a little bit lower than we have with the zonule adherents. But the macula adherents, again, adherents for connecting the cells together, macula for spot-like connection, are going to be also called the desmosomes. And they're like spot welds. And they're going to be there to hold these cells together, to keep the epithelial lining intact. And like the zonule adherents, we need to take the stresses on these connections and distribute it into the cytoskeleton. And what we're going to see with the desmosomes, these macula adherents, is that they're going to distribute the stress into the cytoskeleton by interacting with intermediate filaments. And because we're working with epithelial cells, the primary intermediate filaments that we're going to be dealing with are going to be keratin. But again, it's the same basic idea that we're going to interact with the cytoskeleton and distribute the stress not just on that one point where the membrane's being held together, but distribute it across the entire cell. While we're still on the side of the cells, these lateral specializations, I want to talk about the gap junctions for a minute. The gap junctions are often referred to as a nexus connection or a communicating junction. And basically what it is that the two groups of proteins on adjacent cells line up to one another, and they essentially form a hydrophilic pore. It's like a little docking station. They come together, they open up, and it's going to allow the passage of ions and small molecules to pass from the cytoplasm of one cell into the cytoplasm of the next cell. And so this is going to be important for intercellular communication, for sending signals from one cell to the neighboring cell, so the cells know what's going on. Later on in the course, not dealing with epithelia, but dealing with muscle, we're going to see gap junctions with cardiac muscle because we want to control all of the cardiac muscle cells to contract at the same time. So the presence of the gap junctions in that case, and the muscles in that case, allow ions to flow through and electrical charge to flow through to allow the contraction to be coordinated. Moving down from the lateral side to the base of the cells, we're going to see some basal specializations. And instead of desmosomes, what we're going to have are going to be hemidesmosomes, half desmosomes. So it's going to have the same basic structure, but it's not going to match up with the structure from a neighboring cell. It's basically going to have its half interacting with protein components within the basal lamina. And so it's basically the hemidesmosomes are going to anchor the cell into some of those glycoproteins that are found within that region. And those glycoproteins then are going to be connected up into the connective tissue and anchored to the connective tissue. So we're anchoring the epithelial cell to that underlying tissue. Like the desmosomes, the hemidesmosomes are going to anchor themselves to the keratin in the cytoskeleton to help distribute the stress across the entire cell. Now if we go to the opposite region, Basically, we've got the zonula occludens, and everything lateral and base to that is going to be one domain. Above that ziplock connection is going to be the apical region. And we're going to have some apical specializations that we looked at. The first one are going to be microvilli. Microvilli are going to be finger-like projections of the plasma membrane along that apical surface. And now, in order to support these finger-like projections, we're going to have an actin core. So remember, the properties of actin is that they're nice, rigid molecules that are going to be good for support. And basically, they're going to be there as structural elements to increase the surface area of the apical surface of the cell. So you see lots of microvilli in cells like in the small intestine or the kidney tubules where you've got a lot of absorption occurring. Within the small intestine, it's referred to as a striated border because they're nice and irregular and packed in on one another. In the kidney, it's going to be referred to as the brush border. Similar to the microvilli are going to be stereocilia. Stereocilia aren't true cilia because they have that actin core. They're not going to be motile, but they're again going to be specialized projections found on the apical portion of the cell. Relatively rare within the body. The two primary locations where you can normally find stereocilia are going to be the ductus epididymis within the male reproductive tract and sensory hair cells within the ear. So microvilli are going to be very prevalent within the body, stereocilia a lot less so, but we're also going to have cilia as the third category of these apical specializations. These again are going to be finger-like projections along the apical surface of the cell, but instead of microvilli with an actin core, the cilia are going to have a microtubule core. And the microtubules are going to interact with some of those protein motors that we talked about previously, and they're essentially going to allow these cilia to be motile. So basically the cilia are going to be able to beat in a controlled way, basically in a kind of a wave-like motion, and in doing so, move materials along their surface. And so a lot of times you'll see cilia in regions where you'll have mucus and things that are trapped within the mucus. And so the beating of the cilia, the beating of the waves of the cilia are going to propel the mucus along that epithelial lining. So you can see examples of the cilia in like the respiratory tract, proportions of the respiratory tract, like the trachea and the bronchi. You can see ciliated epithelial cells within the oviducts where the beating of the cilia are going to help propel the egg along the path of the oviduct. And the cilia are going to be similar to flagella, so they're going to be similar to what we're going to see with spermatozoa cells later on in the course. If you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thanks, and I'll see you at part 3.